Malaria is a dangerous and quite often deadly disease. There's medicines for it, but the whole process of recovery is generally uncomfortable and unpleasant, although it beats death by a blood-borne disease. Malaria is caused by the Plasmodium amoeba, with the most insidious and deadly strain being the Plasmodium falciparum. The Plasmodium amoeba is transported through an insect vector, specifically through mosquitoes. The mosquitoes can transfer the amoeba between all of the animals that it bites and takes blood from, and this includes humans. Mosquitoes are the critical vector. As far as humans are concerned, they are the primary threat that carries malaria. Medical research on preventing malaria has taken two broad approaches. The first approach is your typical medical research, wherein we study how it affects the human body and what chemicals can be used to interfere, disrupt, block, or destroy the amoeba inside the human body. The second approach involves the mosquito, wherein we study how malaria behaves in the mosquito's body and what chemicals can be used to interfere with or disrupt that process. As the amoeba has a stage of its life cycle inside the mosquito's body, disrupting this process could destroy the amoeba population, or ideally, at least prevent it from being spread to humans during a mosquito bite. Reducing the mosquito's ability to transmit or carry malaria will inherently lower its risk to humans. It would lower infection rates, and it would reduce overall mortality associated with malaria. The news that I bring you today is a fantastic development in the second type of research approach. As reported by the BBC, a team with members in the UK and Kenya have identified a microbe that has, quote, enormous potential, unquote, to prevent the disease from spreading. So here's the explanation of what this finding is, and how it came to be. In the African nation of Kenya, there's a large body of water in the heart of the Great Rift Valley, called Lake Nyanza, or Lake Victoria. It's the largest lake in Africa, bordered distantly to the east and west by mountains, and more proximally on all sides by gently undulating, sloping lowland, carved with countless streams. Surrounding Lake Victoria is a fertile tropical coastline, running along the riparian zones of the rivers and the streams. This warm, wet, fertile environment presents a heavy insect biodiversity, and within these insects, literally within their bodies, there are countless species of bacteria, and archaea, and protist microbe. Many of these microbes cause diseases, and these insects are their vectors. In one of these insect species, the Anopheles arabiensis mosquito, the scientists discovered a particular microbe that was present in about 5% of the tested individuals. So about 5% of the mosquitoes that they captured and tested had this strange microbe. They named the microbe Microsporidia MB. So first, they say, quote, we show that a vertically transmitted microsporidian symbiont, Microsporidia MB, in the Anopheles gambiae complex can impair Plasmodium transmission, unquote. So in this first part, what they're saying is, when they say vertically transmitted, what this means is that they found a microbe that's passed down from mother to offspring. So once it's infected a lineage, it'll generally stick with that lineage, being passed down through the generations. Furthermore, for the insects that possess this symbiosis, it appears to somehow block the function and the spread of the malaria amoeba. The researchers say, quote, Microsporidia MB is present at moderate prevalence in geographically dispersed populations of Anopheles arabiensis in Kenya, localized to the mosquito midgut and ovaries, and is not associated with significant reductions in adult host fecundity or survival, unquote. Alright, so this is some general background information, and it describes where the microbe exists geographically and where it exists within the host. In this case, it seems to be a gut microbe that also exists in the reproductive structures, in the ovaries, which is presumably how it gets passed on into offspring. And when they say that the microbe is not associated with reductions in host fecundity or survival, this means that for all of the mosquitoes that host Microsporidia MB, the microbe isn't hurting them in any major way. It doesn't cause malformations. It doesn't cause a disease. It doesn't impair their ability to feed and sustain themselves. And most importantly, it doesn't reduce their fertility. It doesn't reduce their chances of mating and producing healthy offspring. Also, this is a method of malaria control that does not kill off the mosquito vector, which means that there shouldn't be any downstream ecological consequences on the birds and the fish and the reptiles that eat mosquitoes as part of their diet. As an aside, a lot of the research on dealing with mosquitoes has involved releasing large quantities of genetically modified mosquitoes, or inoculated mosquitoes, that will reproduce with wild mosquitoes and either fail to produce offspring or will produce offspring that won't hatch. And the whole idea behind this approach is to just kill off the entire population or even the entire species of mosquito that carries the disease. But the problem here is that if you kill off a huge number of mosquitoes, you are potentially taking away a food source for all manner of birds and reptiles and fish and small mammals and whatnot. It should be clarified that there are thousands of species of mosquito, and only several dozen of those species infect humans. And so if you were to kill off all multiple dozens of those species, you would still have hundreds of other mosquito species for these animals to eat. In either case, we'd be rocking the ecological boat, and we'd risk capsizing. And frankly, if we can avoid that risk, we really should. And this Microsporidia MB is just the solution, because it seems to be even more effective at reducing malaria transmission, but it's not fatal. It doesn't involve killing off an entire mosquito species. Anyways, getting back to the paper, the researchers say, quote, field-collected Microsporidia MB infected Anopheles rabiensis tested negative for Plasmodium falciparum gametocytes, and on experimental infection with Plasmodium falciparum, sporozoites aren't detected in Microsporidia MB infected mosquitoes, unquote. Now this part is really cool because they're summarizing their observational and experimental findings, and their findings are awesome. They observed that wild Anopheles rabiensis mosquitoes that hosted Microsporidia MB never seemed to have a simultaneous Plasmodium falciparum infection. And even in their experiment, where they infected these mosquitoes with the Plasmodium falciparum amoeba, the amoeba failed to take root and establish itself. It failed to produce any sporozoites, which are a structure created in this particular life cycle of the amoeba, when it's inside the mosquito host. Because they didn't see any sporozoites in these infected mosquitoes, that would indicate that somehow the presence of this Microsporidia MB is preventing that amoeba infection. How exactly this works is still unknown. The researchers hypothesized that Microsporidia MB could be improving the mosquito's immune system, or it could be secreting some chemical that the malaria amoeba can't tolerate. But more research will be needed to figure out what exactly is going on here. And lastly, the researchers say, quote, as a microbe that impairs Plasmodium transmission that is non-virulent and vertically transmitted, Microsporidia MB could be investigated as a strategy to limit malaria transmission, unquote. This is what they conclude in their paper, so they're basically ending on a hopeful note. They've identified this microbe that can live symbiotically with mosquitoes, which somehow prevents the mosquito from also being infected with malaria. They hypothesized that a malaria prevention mechanism could be achieved if we were to inoculate male mosquitoes and release them into the wild, to potentially create a competitive mosquito lineage that carries the microbe and that also makes up at least 40% of the total mosquito population in the area. If this could be achieved, that would reduce the overall risk they pose of transmitting malaria to humans, and it would save hundreds, if not thousands of lives. But that's a pretty tall order. 40% of the entire mosquito population in the Great Rift Valley of Africa is big. That's a lot of mosquitoes. It's a challenge, but it's well within the realm of technical feasibility. Dr. Jeremy Herron from the International Center of Insect Physiology and Ecology was involved in the research. He was a member of the research team, and he described their findings, saying, quote, the data we have so far suggests it is 100% blockage. It's a very severe blockage of malaria, unquote. This really is a breakthrough development, and I'm excited to see where the research will take us in the near future. Even if this method doesn't work that well on other malaria strains or in other species of mosquito, it's still incredible. What matters most is that it works in Anopheles rabiensis and it works on Plasmodium falciparum. This matters a lot because Anopheles rabiensis is one of the most notorious mosquito species for spreading diseases, and it matters because Plasmodium falciparum is the most deadly malaria strain. So even if Microsporidia MB doesn't work at all in any other mosquito species, and even if it doesn't do anything at all to any other malaria strain besides Plasmodium falciparum, this is still a really big win for the human species. This is really good news, and it could realistically open a door to a future where malaria is a thing of the past. And that would be a beautiful future for all mankind.